Hello, everyone. Welcome back to Decoding Dynasty, where we unravel the fascinating intersections of history and genetics. In our previous episodes, we've explored the genetic legacies that have shaped royal families. Today, we turn our attention to a figure recently brought to vivid life on screen in Queen Charlotte, a Bridgerton story. King George III, the monarch who reigned over Great Britain during a pivotal era spanning the late 18th and early 19th centuries. As many viewers of the series will know, King George experienced periods of profound and unsettling illness, often referred to as the royal malady. These episodes of severe mental and physical distress were not just a personal tragedy for the king and his family. They also had significant political and social ramifications for the nation. For centuries, the true nature of King George III's ailment has been a subject of intense historical and medical debate. Numerous diagnoses have been proposed, ranging from psychological distress to physical ailments. Among the most compelling and enduring theories, particularly in the late 20th century, was the suggestion that the king suffered from a rare inherited disorder called Porphyria, affecting the body's ability to process hem. This theory gained traction based on retrospective analyses of his reported symptoms, which included episodes of abdominal pain, discolored urine, and marked neuropsychiatric disturbances, along with accounts of similar issues in some of his relatives. However, more recent and sophisticated analyses, utilizing modern medical knowledge and even computer-based diagnostic tools, have increasingly pointed towards a different explanation: bipolar disorder, also known as manic depressive illness. These studies argue that the cyclical nature of King George's manic episodes, his periods of apparent recovery, and his eventual cognitive decline align more closely with this psychiatric diagnosis. Today, we'll delve into the historical context of King George III's royal malady and explore these competing theories. While current scientific consensus leans towards bipolar disorder, the Porphyria hypothesis remains a significant part of the historical narrative. We'll then take a closer look at what Porphyria actually is, its genetic underpinnings, its biochemical pathways, and the array of symptoms it can cause, much like we did with hemophilia in our previous episode. Join me as we decode the complex health situation of King George III and the science behind the Porphyria theory. What are Porphyrias? At its core, Porphyria refers to a collection of inherited or, in some instances, acquired metabolic disorders stemming from defects in the heme biosynthetic pathway. Heme, an iron-containing porphyrin ring, is a crucial component of numerous essential proteins, most notably hemoglobin in red blood cells, cytochromes involved in cellular respiration, and various other enzymes critical for life. The synthesis of heme is a meticulously orchestrated 8-step enzymatic process, each step under precise genetic control. In Porphyrias, a deficiency or malfunction in any one of the genes encoding these enzymes leads to the pathological accumulation of specific heme precursors, porphyrins or their biochemical predecessors, in various tissues, including the liver, bone marrow, and erythrocytes. These accumulated intermediates are often toxic, giving rise to a diverse array of clinical manifestations. Historically, Porphyrias have captured the imagination, with some speculating their role in historical figures exhibiting unusual behaviors. However, our understanding today is firmly rooted in molecular genetics and biochemistry. Clinical symptoms of Porphyrias. The clinical spectrum of Porphyrias is remarkably broad, reflecting the specific gene mutation, the affected enzyme, the site of precursor accumulation, and the nature of the accumulated compounds. We broadly categorize Porphyrias based on their primary site of metabolic dysfunction: hepatic Porphyrias, where the liver is the main site of precursor overproduction, and erythropoietic Porphyrias, where the bone marrow and red blood cells are primarily affected. Hepatic Porphyrias often manifest with acute, potentially life-threatening attacks characterized by severe abdominal pain, frequently described as colicky and poorly localized. These attacks can be accompanied by gastrointestinal disturbances such as nausea, vomiting, and constipation. Critically, many acute hepatic porphyrias also involve the nervous system, leading to a range of neurological symptoms, including peripheral neuropathy, muscle weakness, pain, and sensory changes, seizures, and even encephalopathy with confusion, hallucinations, and altered mental status. Autonomic nervous system dysfunction can result in tachycardia, hypertension, and bladder dysfunction. In contrast, erythropoietic porphyrias primarily present with cutaneous photosensitivity. Exposure to sunlight, particularly in the visible light spectrum, can cause painful, non-blistering skin reactions, itching, swelling, and chronic skin changes. Some erythropoietic porphyrias can also lead to anemia and splenomegaly. It's important to note that some porphyria types can exhibit a combination of both acute neurovisceral and cutaneous symptoms. The variability in clinical presentation underscores the complexity of these disorders and the need for careful diagnostic evaluation, often guided by genetic insights. Genetics of porphyrias. The vast majority of porphyrias have a strong genetic basis, resulting from inherited mutations in the genes encoding the 8 enzymes of the heme biosynthetic pathway. These genes, along with their chromosomal loci, are ALAS1, aminolevulinate synthase 1, located on chromosome 17q21.33. This gene primarily functions in the liver and other non-erythroid tissues. AAD, aminolevulinate dehydratase, located on chromosome 9q34. This gene encodes a cytosolic enzyme expressed in all tissues. HMBS, hydroxymethylbilane synthase, also known as PBGD, porphobilinogen deaminase, located on chromosome 11q23.3. This gene encodes a cytosolic enzyme expressed in all tissues, with erythroid-specific and non-erythroid-specific promoters. UROS, uroporphyrogen synthase, located on chromosome 1026.3. This gene encodes a cytosolic enzyme expressed in all tissues. UROD, uroporphyrogen decarboxylase, located on chromosome 1p34.1. This gene encodes a cytosolic enzyme expressed in all tissues. CPOX, coproporphyrogen oxidase, located on chromosome 3q12.1, q12.2. This gene encodes a mitochondrial enzyme expressed in all tissues. PPOX, protoporphyrogen oxidase, located on chromosome 1q23.3 to q24.1. This gene encodes a mitochondrial enzyme expressed in all tissues. FEC, ferrochelatase, located on chromosome 18q21.3. This gene encodes a mitochondrial enzyme expressed in all tissues, with higher levels in erythroid cells. ALAS2, aminolevulinate synthase 2, located on chromosome XP11.21. This gene is specifically expressed in erythroid cells. Over 1,000 different mutations have been identified across these genes, contributing to the significant molecular heterogeneity observed in porphyrias and the wide spectrum of clinical presentations. These mutations, ranging from single nucleotide polymorphisms (SNPs) leading to missense or nonsense codons, to insertions, deletions, and splice site alterations, each affect enzyme activity and expression to varying degrees. Understanding the specific genetic defect and its location on the genome is paramount for accurate diagnosis and predicting inheritance patterns. Ongoing research is crucial to differentiate between truly pathogenic variants and benign genetic polymorphisms, refining our diagnostic accuracy and informing genetic counseling. The inheritance patterns of porphyrias vary, reflecting the nature of the mutated gene and its chromosomal location. The acute hepatic porphyrias, namely acute intermittent porphyria, AIP, due to mutations in HMBS 11q23, hereditary coproporphyria, HCP, due to mutations in CPOX 3q12.12.2, and variegate porphyria, VP, due to mutations in PPOX 1q23 3q24.1, are typically inherited in an autosomal dominant manner. This means that a single copy of the mutated allele from one parent is usually sufficient to predispose an individual to the condition. However, these conditions often exhibit low penetrance, meaning that not all individuals carrying the pathogenic genotype will develop clinical symptoms. The reasons for this variable expressivity and penetrance are complex and likely involve modifier genes and environmental factors. Rarer, more severe homozygous dominant forms, where an individual inherits 2 copies of a severely mutated allele, can also occur. In contrast, some porphyrias, such as ALA dehydratase deficiency porphyria (ADP) due to mutations in ALADE 9q34, congenital erythropoietic porphyria (CE) due to mutations in UROS 10q26.3, and erythropoietic protoporphyria (EPP) due to mutations in FH 18q21.3, follow an autosomal recessive inheritance pattern. In these cases, an individual must inherit 2 copies of the mutated allele, one from each parent, to manifest the disease. These conditions often present with more severe phenotypes due to the complete or near-complete loss of functional enzyme activity. There are exceptions to these typical Mendelian patterns. X-linked protoporphyria, XLP, a subtype of erythropoietic protoporphyria, is caused by gain-of-function mutations in the ALS2 gene located on the X chromosome, XP11.21. This leads to increased activity of erythroid-specific ALA synthase. Furthermore, Porphyria cutanea tarda, PCT, the most common porphyria, demonstrates complex genetics. While there are familial forms with inherited mutations in the UROD gene, 1p34.1, autosomal dominant with low penetrance, the sporadic form is more prevalent and involves a combination of genetic predisposition, often a UROD polymorphism, and acquired factors such as iron overload and hepatitis C infection. Understanding the precise genetic defect and inheritance pattern through molecular genetic testing is crucial for accurate diagnosis, genetic counseling, and risk assessment for family members, allowing for informed reproductive decisions and potential preventative measures. Pathogenetics of porphyrias. The pathogenesis of porphyrias involves a complex interplay of underlying genetic defects, the resulting biochemical consequences, and various environmental influences. The primary pathogenic mechanism is the tissue-specific accumulation of specific heme precursors proximal to the defective enzyme in the biosynthetic pathway. The specific gene mutation dictates which enzyme, encoded by genes on specific chromosomal loci as we just discussed, is affected, and consequently, which specific porphyrins or porphyrin precursors build up. Whether the liver in hepatic porphyrias or the bone marrow in erythropoietic porphyrias is the primary site of this accumulation largely determines the predominant clinical manifestations and the types of accumulated metabolites detected in diagnostic testing. The metabolite accumulation itself is toxic through various mechanisms. For instance, in acute hepatic porphyrias, the buildup of 5-aminolevulinic acid, ALA, and porphobilinogen, PBG, is believed to exert direct neurotoxic effects. ALA, structurally similar to the inhibitory neurotransmitter GABA, has been shown to interact with GABA receptors in the brain, potentially disrupting normal inhibitory neurotransmission and contributing to anxiety, seizures, and other neurological disturbances. Furthermore, ALA can directly damage nerve cells through mechanisms involving oxidative stress and excitotoxicity. PBG, while its precise neurotoxic mechanism is less well defined, is also thought to play a significant role in the neurological syndrome of acute porphyria. In the cutaneous porphyrias, the accumulated porphyrins, such as uroporphyrin and coproporphyrin, are highly photosensitive. Upon exposure to specific wavelengths of light, these molecules become excited and transfer energy to molecular oxygen, generating highly reactive oxygen species that cause direct damage to skin cells, leading to the characteristic photosensitivity and skin lesions. The severity and penetrance of porphyrias are not solely determined by the primary genetic defect, the specific mutation on a particular chromosomal locus. Modifier genes, which are variations in other genes within an individual's genome, located on various chromosomes, can influence the expression and severity of the disease by affecting enzyme stability, metabolic flux, or detoxification pathways. Furthermore, various environmental factors can act as triggers or exacerbating factors, particularly in individuals with an underlying genetic predisposition. These include exposure to certain drugs, especially cytochrome P450 inducers that increase heme turnover and thus the flux through the pathway, alcohol consumption, smoking, infections that increase metabolic demand, hormonal fluctuations, especially in women, and their link to acute attacks, and nutritional status, such as periods of fasting or calorie restriction. In PCT, the interplay between a genetic predisposition, often a hypomorphic urode allele on chromosome 1p34.1, and acquired factors like iron overload, which directly inhibits the already compromised uroporphyrinogen decarboxylase enzyme, and hepatitis C infection, which can also impact liver function, is critical for disease manifestation. The complex interplay of these genetic at specific genomic locations and environmental factors contributes to the highly variable clinical course and expression observed in porphyrias, making accurate diagnosis and personalized management essential. Biochemistry of porphyrias. The biochemistry of porphyrias is fundamentally rooted in the disruption of the precisely regulated 8-step heme biosynthetic pathway. This pathway, occurring in both the mitochondria and the cytoplasm of cells, begins with the condensation of succinyl-CoA from the Krebs cycle and glycine, catalyzed by the enzyme ALA synthase ALAS, with ALS1, encoded by a gene on chromosome 7q2133, being the predominant form in most tissues, and ALS2, encoded by a gene on chromosome XP11.21, being erythroid-specific. Subsequent enzymatic steps involve a series of complex modifications, including the condensation of 2 molecules of ALA by ALA dehydratase, ALAD, chromosome 9q4, to form porphobilinogen, PBG, the polymerization of 4 PBG molecules by hydroxymethylbilane synthase, HMBS, chromosome 11q23.3, also known as porphobilinogen deaminase, to form hydroxymethylbilane, and the cyclization and modification of this linear tetrapyrrole by uroporphyrinogen synthase, UROS, chromosome 10q26.3, and uroporphyrinogen decarboxylase, UROD, chromosome 1p34.1, to yield coproporphyrinogen 3. This molecule then enters the mitochondria, where coproporphyrinogen oxidase, CPOX, chromosome 3q12.1q12.2, and protoporphyrinogen oxidase, PPOX, chromosome 1q23.3 to q24.1, catalyze further oxidations to form protoporphyrin 9. Finally, the enzyme ferrochelatase, ECH, chromosome 18q to 1.3, incorporates ferrous iron into protoporphyrin 9 to produce heme. In each specific type of porphyria, a genetic deficiency due to a mutation on a specific chromosomal locus leading to reduced activity of one of these 8 enzymes creates a metabolic bottleneck in the pathway. Consequently, the heme precursors that are substrates for the deficient enzyme accumulate in the affected tissues, liver or bone marrow, while the production of heme and subsequent downstream metabolites is reduced. These accumulated precursors are then thought to be diverted into alternative metabolic pathways, leading to the production of abnormal porphyrins and other compounds that are implicated in the pathophysiology of the acute neurovisceral attacks. The specific pattern and concentration of accumulated porphyrins and their precursors in biological fluids, urine, plasma, and feces are often pathognomonic for each type of porphyria. Quantitative biochemical analysis of these markers is therefore a cornerstone of diagnosis, allowing for the identification of the specific enzymatic defect, often inferring the affected gene and its locus, and the classification of the porphyria subtype. Furthermore, monitoring these biochemical markers can be valuable in assessing disease activity and the response to treatment. Cellular and molecular pathways in porphyrias. At the cellular and molecular level, the genetic defects in porphyrias trigger a cascade of downstream effects that extend beyond the immediate disruption of heme biosynthesis. The accumulation of heme precursors can induce significant oxidative stress within affected cells and tissues. For instance, ALA has been shown to stimulate the production of reactive oxygen species, ROS, leading to damage of cellular macromolecules such as lipids, proteins, and DNA. This oxidative damage is thought to contribute to both the neurological and hepatic manifestations of porphyrias. Furthermore, these accumulated precursors can directly interact with various cellular signaling pathways. ALA, for example, has been implicated in modulating the gamma-aminobutyric acid, GABAergic neurotransmitter system in the central nervous system. By potentially acting as a GABA agonist or by interfering with GABA metabolism, ALA may contribute to the neurological and psychiatric symptoms observed during acute porphyria attacks. The deficiency of heme itself, resulting from the genetic defect in one of the heme biosynthesis genes, also has far-reaching consequences. Heme is an essential prosthetic group for a wide array of vital enzymes, including the cytochrome P450 enzyme superfamily in the liver, which are crucial for the detoxification of drugs and other xenobiotics. Reduced heme levels can impair the function of these CYP enzymes, potentially exacerbating drug-induced porphyria attacks. Heme is also critical for mitochondrial respiratory chain complexes involved in energy production. In some porphyrias, impaired mitochondrial function has been observed, potentially contributing to cellular dysfunction and disease symptoms. In the liver, the chronic accumulation of porphyrins can trigger inflammatory responses, leading to the release of pro-inflammatory cytokines and the activation of fibrogenic pathways involving hepatic stellate cells. Over time, this chronic inflammation and fibrosis can progress to cirrhosis in certain porphyria types. In erythropoietic porphyrias, the accumulation of highly photosensitive porphyrins within erythrocytes leads to increased red blood cell fragility and premature destruction, contributing to hemolytic anemia. Understanding these complex cellular and molecular pathways that are perturbed in porphyrias as a consequence of genetic defects at specific chromosomal locations is not only crucial for elucidating the full pathophysiology of these disorders, but also for identifying potential targets for therapeutic interventions aimed at mitigating the downstream consequences of the primary genetic defect. Neurogenetics of porphyrias. The neurogenetic aspects of porphyrias, particularly the acute hepatic forms, AIP due to HMBS on 11q20.3, HCP due to CPOX on 312.1 to Q12.2, VP due to PPOX on 1q23.3, Q24.1, represent a critical area of investigation in understanding the debilitating neurological manifestations associated with these genetic disorders. The involvement of the peripheral, central, and autonomic nervous systems is a hallmark of acute attacks, and unraveling the underlying mechanisms of neurological dysfunction is essential for developing effective treatments. Elevated levels of both ALA and PBG in the peripheral nervous system can lead to a predominantly motor axonal neuropathy, often presenting with progressive muscle weakness that can mimic Guillain-Barré syndrome. Sensory involvement may also occur, and autonomic nerve dysfunction is common, manifesting as severe abdominal pain, likely due to visceral neuropathy, tachycardia, hypertension, constipation, and bladder dysfunction. Beyond the direct neurotoxicity of ALA and PBG, metabolic disturbances within the nervous system may also contribute to the neurological manifestations. Alterations in the metabolism of tryptophan, a precursor to serotonin and melatonin, have been observed in acute porphyria, potentially contributing to neuropsychiatric symptoms such as anxiety, insomnia, and mood changes. Additionally, the reduced availability of heme in neural tissues may impair the function of heme-dependent enzymes crucial for neuronal metabolism and function. In severe acute attacks, particularly those with significant central nervous system involvement like seizures and encephalopathy, cerebral vasoconstriction and brain edema, sometimes culminating in posterior reversible encephalopathy syndrome, have been increasingly recognized as contributing factors. The complex interplay of these genetic, at specific genomic locations, biochemical, and neurological factors underscores the importance of a multidisciplinary approach to the diagnosis and management of the neurological complications of acute porphyrias. Genetic factors may also influence the susceptibility and severity of neurological involvement, although the specific modifier genes on various chromosomes are still being investigated. The central role of the liver in porphyrias. While the primary genetic defect in porphyrias is systemic, affecting the potential function of heme biosynthetic enzymes in all tissues, the liver plays a particularly central role in the pathogenesis of many types, most notably the hepatic porphyrias. As the principal site of heme synthesis for the production of essential hemoproteins, like cytochrome P450 enzymes involved in drug metabolism and detoxification, the liver is often the primary location where the enzymatic bottlenecks in the heme pathway lead to the most significant accumulation of toxic heme precursors. The acute hepatic porphyrias, including acute intermittent porphyria, AIP, hereditary coproporphyria, HCP, and variegate porphyria, VP, as well as the more common Porphyria cutanea tarda, PCT, are directly linked to genetic deficiencies of specific enzymes expressed predominantly in the liver. In AIP, for instance, the genetic mutation in the HMBS gene results in a deficiency of hydroxymethylbilane synthase activity within hepatocytes. This enzymatic block leads to a substantial intrahepatic accumulation of the neurotoxic precursors ALA and PBG. The activity of the first and rate-limiting enzyme in the hepatic heme pathway, ALA synthase 1, is normally under negative feedback control by heme. In the setting of a downstream enzymatic deficiency, the reduced hepatic heme levels lead to a de-repression and increased activity of ALAS1. This increased flux through the initial steps of the pathway further exacerbates the accumulation of the problematic heme precursors. Factors that induce ALAS1 expression in the liver, such as certain drugs, cytochrome P450 inducers that consume heme, fasting, and hormonal fluctuations, can therefore dramatically amplify the buildup of these toxic intermediates and trigger acute porphyria attacks in genetically susceptible individuals. The chronic intrahepatic accumulation of porphyrins and their precursors can also have direct hepatic consequences. In some porphyria types, such as erythropoietic protoporphyria, EPP, where protoporphyrin IX, although primarily produced in the bone marrow, accumulates in hepatocytes and bile canaliculi, this can lead to cholestasis, liver inflammation, fibrosis, and, in severe cases, progressive liver damage and liver failure. Alarmingly, patients with hepatic porphyrias, particularly the acute forms, have a significantly increased risk of developing hepatocellular carcinoma HCC, even in the absence of pre-existing cirrhosis. The mechanisms underlying this increased cancer risk are thought to involve chronic oxidative stress induced by the accumulated porphyrin precursors and potentially direct genotoxic effects. Moreover, studies have indicated that active hepatic porphyria can be associated with impaired mitochondrial function within hepatocytes, potentially contributing to reduced cellular energy production and further impacting liver function and overall disease severity. Diagnosis and screening in porphyrias. Accurate and timely diagnosis and screening are paramount in the management of porphyrias, enabling prompt treatment, prevention of debilitating complications, and the identification of at-risk family members who may carry the causative gene mutation. Given the rarity and the diverse clinical presentations of these disorders, a systematic diagnostic approach is essential. The diagnostic process typically begins with stepwise biochemical testing guided by the patient's presenting symptoms. Number 1. For acute neurovisceral symptoms, a rapid screening test for urinary porphobilinogen, PBG, is highly recommended due to its characteristic elevation during acute attacks of hepatic porphyria. For cutaneous symptoms, initial investigations include the analysis of plasma or erythrocytic porphyrins and urinary porphyrins, specifically in cases of blistering skin lesions. Positive screening results necessitate further targeted quantitative testing and, if required, more detailed analysis of porphyrin profiles in urine, plasma, and fecal samples to definitively identify the specific type of porphyria and infer the likely affected gene. It is crucial that all biological samples are meticulously protected from light to prevent the degradation of the light-sensitive porphyrins and their precursors, ensuring accurate results. Validated laboratory diagnostic algorithms based on presenting symptoms have demonstrated high sensitivity and specificity for both acute and cutaneous porphyrias, aiding clinicians in the complex interpretation of laboratory findings. Number 2. Molecular genetic testing, involving DNA analysis of the genes involved in heme biosynthesis, plays a vital role in confirming the specific genetic mutation in affected individuals. Importantly, genetic testing is the only reliable method for screening asymptomatic gene carriers within families. Identifying the specific pathogenic variant on its chromosomal locus allows for precise genetic counseling. However, it's crucial to understand that genetic testing alone does not indicate the current disease activity or severity. In some cases, particularly when genetic results are inconclusive or unavailable, the measurement of specific enzyme activities in relevant tissues, erythrocytes for ELAD, HMBS, UROS, FECH, and lymphocytes for urode, CPOX, PPOX, and liver biopsy in rare cases can provide supportive diagnostic evidence, often correlating with the expected impact of the identified gene mutation. Once a porphyria diagnosis is confirmed in an individual through a combination of biochemical and genetic testing, identifying the specific gene and its location, family screening for the familial mutation is essential to identify presymptomatic carriers. This allows for informed genetic counseling, providing individuals with a clear understanding of their risk and enabling proactive strategies to avoid known precipitating factors, which is particularly critical in the acute hepatic porphyrias. Early and accurate diagnosis, guided by both biochemical and genetic findings, is of paramount importance, as prompt treatment can significantly mitigate the severity of attacks, prevent long-term complications, and reduce morbidity and mortality, especially in the potentially life-threatening acute porphyrias. Due to the complexity of these disorders, patients with confirmed or suspected porphyria should be referred to specialized porphyria centers for comprehensive management and ongoing follow-up care. Treatments for porphyrias. The treatment of porphyrias is highly individualized, depending on the specific type of porphyria, determined by the affected gene locus, the severity of symptoms, and whether the patient is experiencing an acute attack or chronic manifestations. For acute hepatic porphyrias, the immediate management of an acute attack revolves around hemin therapy, or heme arginate. Intravenous administration of hemin helps to repress hepatic ALA synthase 1, thereby reducing the overproduction of the neurotoxic precursors ALA and PBG and alleviating the acute symptoms. In milder attacks, high-dose intravenous glucose may be considered, but hemin is the preferred and more effective treatment for moderate to severe episodes. Comprehensive supportive care, including hospitalization, meticulous withdrawal of any potential trigger drugs, careful correction of electrolyte imbalances, effective pain management, and ensuring adequate nutrition with safe medications is crucial during acute attacks. A significant advancement in the treatment of acute hepatic porphyrias is therapeutic RNA interference, RNAi. Givosiran is an RNAi therapeutic that specifically targets and reduces the expression of the ALAS1 gene in the liver. By inhibiting ALAS1, Givosiran effectively prevents the excessive production of ALA and PBG. Clinical trials have demonstrated remarkable efficacy, with substantial reductions in the frequency of acute attacks and a normalization of the levels of these toxic metabolites. Givosiran has generally shown a manageable safety profile, representing a major step forward in the long-term management of these debilitating conditions. The development of other emerging therapies, such as gene therapy, aiming to correct the underlying genetic defect on the respective chromosome, innovative mRNA-based enzyme replacement strategies, and the use of molecular chaperones to help stabilize defective enzymes, holds further promise for the future treatment of acute porphyrias. The treatment of cutaneous porphyrias focuses on reducing porphyrin accumulation in the skin and minimizing photosensitivity. For porphyria cutanea tarda, PCT, the primary approaches involve reducing iron overload through regular phlebotomy or using low-dose antimalarial drugs like hydroxychloroquine or chloroquine to enhance porphyrin excretion. Addressing underlying conditions that can exacerbate PCT, such as hepatitis C or excessive alcohol consumption, is also essential. In erythropoietic protoporphyria, EPP, strict avoidance of sunlight and the use of protective clothing and sunscreens are paramount. Oral beta-carotene can sometimes offer some photoprotection. In severe cases of EPP with liver involvement, liver transplantation may be considered. For the rare and severe congenital erythropoietic porphyria, allogeneic hematopoietic stem cell transplantation is the only curative option for severe cases. Long-term management across all porphyria types emphasizes the avoidance of known precipitating factors. Educating patients about these triggers, which can include specific medications, alcohol, fasting, and infections, is crucial. Genetic counseling and family screening, identifying carriers of the specific gene mutation on its chromosomal location, play a vital role in identifying at-risk individuals and implementing preventive strategies. As our exploration of porphyrias on Decoding Dynasty draws to a close, we've uncovered the intricate genetic and biochemical disruptions in the vital heme synthesis pathway, tracing them to specific locations within our genome. These genetic variations can lead to a wide spectrum of symptoms, highlighting the power of our genes in shaping our health. It's fascinating to consider historical figures like King George III, whose perplexing episodes sparked centuries of speculation, with Porphyria emerging as a potential explanation. This historical link underscores the long and often mysterious history of these genetic metabolic diseases. Today, thanks to advances in genetics and molecular biology, we are moving beyond historical enigmas. Precise genetic diagnoses and innovative treatments, such as RNA interference, are offering new hope for individuals living with porphyria. Continued research and increasing awareness promise a brighter future for those affected. If you found this deep dive into the genetics of porphyrias insightful, please take a moment to like this episode. We'd also love to hear your thoughts or any questions you might have in the comment section below. And if you're eager to delve even deeper into the research behind topics like this, be sure to check out my Telegram channel, where I share the articles I've researched for each episode. Finally, to stay up to date on all our genetic explorations, please subscribe to my channel so you don't miss future episodes. Thank you for joining me on this episode. Until our next genetic exploration, stay curious.